Welcome back to another episode of Human Biology with Dr. B. Today we're finishing up our talk on the cell. We're going to look at movement into and movement out of the cell today. We're going to talk about diffusion. Now, we've talked about this in lab already, but the basic definition of diffusion is movement of a substance from a higher concentration to a lower concentration. So what does that mean? That means that we have, as I drew in class, here's our slide. We're going to picture a toddler at the top of the slide. Now, the toddler at the top of the slide has a lot more energy than when the toddler gets to the bottom of the slide. So if the toddler, one of the reasons that we as parents freak out when we see a tiny little baby up here is because we're like, oh no, she's going to come down so quickly. It's the same thing with diffusion. When there's a high concentration of molecules in one area, it naturally wants to move to be at a more equal distribution throughout what we call the solution. And there are two types. We have simple diffusion and we have carrier-mediated diffusion. Here in this picture, you can see that we have two beakers, if you will, and a semi-permeable membrane in the middle. And if we allow time and what we call Brownian motion, the natural movement of the molecules over time, the molecules will become equally distributed throughout the solution and across the membrane. You can see here we have our plasma membrane, our phospholipids right here are with the phosphate head and the lipid tails, the lipids forming what we call the phospholipid bilayer and a very semi-permeable plasma membrane. So we see on the extracellular surface we have a high concentration of these blue molecules, and then on the cytoplasm we have a low concentration of these blue molecules. Now, the blue molecules naturally want to be able to move from high to low. So what does this mean? That means that they need to be able to move through the plasma membrane. And we're going to talk about different ways that this can happen. Then we get to carrier-mediated transport. We have two different types. We have passive carrier-mediated transport and we have active carrier-mediated transport. Passive carrier-mediated transport is what we call facilitated diffusion. As you can see in this picture, we have proteins embedded, we call these integral proteins, embedded in the membrane that allow for molecules of a high concentration external to the cell to move into the cell so that the concentration is even on either side of the plasma membrane. Now we also have active diffusion. Now active diffusion is when energy is expended in order to move the molecule against its concentration gradient. So that means that it goes up the slide. Now, if you've ever tried to walk up a slide or watched your child in socks try to get up the slide, you know that it takes more energy to physically climb the slide than it does to just sit at the top and whee down. This is the difference between diffusion or facilitated diffusion and active transport. Active transport uses energy to move against that concentration gradient. In either case, we need an integral protein to be used as a transport molecule. Now, any type of integral protein is specific for one type of molecule. As you see here in this picture, this is the glucose transporter that moves it across the membrane. And this protein will only bring glucose molecules across the membrane. Now, very similar to enzymes, these proteins can become saturated as well as they can be regulated as to how much is let in and how much is removed out using diffusion or using expending energy. Now, active transport, as we've talked about, it requires ATP to pump against the concentration gradient. As I mentioned, it doesn't depend on the concentration gradient, whereas diffusion, we need a high concentration gradient, and with this high concentration gradient, there's a sort of energy that allows for movement of the molecules so that it's evenly distributed. Now, the most common here is the ion pump. As you can see in this picture, we have the sodium-potassium pump. This is the pump right now that's allowing you to continue to fire neurons in order to remember and understand this material. We'll get to that when we get to the neurological system. But here we go. So here on the inside, we have sodium ions, 1, 2, 3. They come in and they bind in the sodium-potassium pump. Now, these are released external to the cell and then 2 potassiums come in, 1, 2, and they bind to this particular pump and are released inside. Now what this allows to happen is for the sodium concentration to increase on the outside and the potassium concentration to increase on the inside. This plays a major role in any type of what we call an action potential or neuron firing in your neurological system. And here you can see another example of active transport. Here we have a high concentration inside the cell, and we're still going to attempt to pump more into the cell. So we use ATP, we cleave a phosphate so that we make it into ADP, and this will move a solute against the concentration gradient into the cell, from low to high. Now, osmosis is diffusion referring only to the movement of water molecules, and this is very specific because it's across a semi-permeable membrane. Osmosis never occurs on its own, it's always in reference to a semi-permeable membrane. Now, in this instance, the membrane is only permeable to water and not to solutes. As you can see here, we have a semi-permeable membrane across the U-tube. This is the original YouTube. It's not the YouTube on which you're watching this. This is the chemistry YouTube. And what happens is this membrane is only permeable to water molecules. And as you can see, initially on the left side, there is a higher concentration on the left side than there is on the right side. Now, because the pressure and the energy of the movement of these molecules wants an even concentration distribution across the semi-permeable membrane, the water will physically move so that way it has an even number of water molecules on one side, an even number of solute molecules on one side and on the other side. Which brings us to the discussion of tonicity. There's 3 basic types of tonicity we need to be aware of: hypo, hyper, and iso. Now, anytime we discuss tonicity, it is in relation to something else. So, for example, if we were to put a cell in a solution, we would define the tonicity with regard to the cell or the solution. So, work with me here. A hypotonic solution, think of the word hypo. Usually when we think of the word hypo, we think of hypoglycemia, we're about to crash, we get hangry, etc. Hypo means there's not as much. Then we take the prefix hyper. Hyper. Now, if you have ever encountered a preschooler or a kindergartner or a first grader or even just any young child on sugar on Halloween, then you are well aware of what hyper means. It means a lot. In this case, a lot of sugar equals a lot of energy in these children. When we talk about tonicity, it means more solute. And then isotonic means that it is the same. Now, when we discuss a cell in relation to a solution, we can talk about the cell in a hypertonic solution, as you see here on the left. If we put a cell in a hypertonic solution, that means there is more solute molecules outside of the cell. And where does the water want to go? The water wants to evenly distribute across the membrane according to the solute molecules. So the water will go out of the cell. And we call this crenation. Now, over here on the right, we have cells in a hypotonic solution. That means that there are more solutes in the cell than in the solution. So in this case, we're describing the tonicity of the solution with regard to the cell. So less solutes in the water, more in the cell. So in this case, what's going to happen? Where's the water going to want to go? The water is going to want to go into the cell to, again, help distribute the concentration of solutes, to make them even across the membrane. In this case, water will go in and the cell will burst. We call this plasmolysis, or if we're talking about red blood cells, we call this hemolysis. Now, if a cell is in an isotonic solution, nothing happens. It's happy, it's wonderful. And this is exactly why we don't ever hook a sick person up to an IV and just turn the faucet on. Because the water in the tap would be hypotonic to your cells. Your cells in your blood, if you ever look at the saline solution that they put in, I believe it's, I don't know, like 0.07% salt. It also can have some other things in it, but this is the same concentration of solutes that are in your blood cells. So yes, if you were to inject yourself with just regular water, it would cause massive problems in your body. Probably throw some clots, burst some cells, pretty ugly all the way around. Now, here is another depiction of this. As you can see here on the left, we have this is just like what we worked with in lab. It is a dialysis tubing bag in which we have a 98% water, 2% sugar solution, so not a lot of sugar. However, if we put it in a hypertonic solution, that means that there is more sugar in the solution than in the bag. And in this case, the water would go out. So that, because the goal of any type of energy that these molecules have as they move is because they want to have an even distribution across the membrane. So if there's less sugar inside and more outside, that means the water within the bag or the cell is going to leave. Isotonic, there's even flow of molecules into and out. Everything's happy-go-lucky. And then if we were to put this in a hypotonic solution, that means there are less solutes in the solution than in the cell or the bag. And that means the water is going to rush in order to evenly distribute, to have an even distribution of solutes across the membrane. Which brings us to large movement of molecules across the membrane. Have you ever wondered how these bacterium, look at those hairy little things, get into your white blood cells and how your white blood cells kill them? This is how they do it. They do it by endocytosis. Now, specifically, we call this phagocytosis. This is cell eating. This is where a cell uses its pseudopodia. That's what these guys are right here. Pseudopodia means fake feet. And the cell membrane basically comes up and around a bacteria or a larger molecule. Anytime we're dealing with this, cell eating is phagocytosis. It moves in, creates a vesicle, and in the case of white blood cells, this phagosome, as you can see here with the bacteria in it, will then merge with a lysosome, which we just finished talking about in the previous lecture. And the lysosome will help digest the bacteria and break it up so that it no longer can harm the body or the organism. And then we have pinocytosis. Pinocytosis is cell drinking. This is drinking. And typically in cell drinking, this is where larger volumes of small molecules and extracellular fluid are moved into the cell. Again, it's the same thing. We have pseudopodia. These are the pseudopodia right here. They move out and around some of the extracellular fluid, bringing in some solutes. It forms a vesicle, and this vesicle will go to where it's needed within the cell. And then we have exocytosis. Exocytosis moves in this about the same way that endocytosis does, except it's in reverse. As you can see here, step 1, the vesicle moves through the cytoplasm towards the cell membrane. It will then fuse with the cell membrane, and as the membrane fuses, all the contents are released into the extracellular fluid. Now, this is how proteins that are made on the rough endoplasmic reticulum, which we just talked about, move through the rough ER to the Golgi to the cell membrane for excretion. They do it through exocytosis. And last but not least, we're talking about metabolism. Yet again, we can't stress this enough. Now, metabolism is all the chemical reactions that take place within a cell. It is the sum of all the anabolic and catabolic chemical reactions within a cell. Now, one of the main things that we need to talk about here is cellular respiration. And this is how cells produce ATP. Remember, ATP is the energy currency of life. And so ATP needs to be produced from the macromolecules, either ingested by phagocytosis, endocytosis. We just saw how glycolysis comes in through carrier-mediated transport. So how are all these molecules broken down and basically made into ATP so that the cells can have energy? Well, they do it in 3 steps. Cellular respiration has 3 major steps. First one is glycolysis, where we split a molecule of glucose. Then we go to the Krebs cycle inside the mitochondrion, and then the electron transport chain. And here's a great picture of all of this. So number 1, so here we have this, putting it all together. So here we have the glucose molecule. It moves inward through a carrier protein. The glucose in the cytoplasm, in the cytoplasm, the glucose is then, through a series of chemical reactions, broken down into pyruvate. So glucose is a 6-carbon sugar, pyruvate is a 3-carbon sugar. So they basically make 2 3-carbon sugars. Now pyruvate then goes into the matrix of the mitochondrion. Remember, it's the powerhouse of the cell, and here's why. It does a transition reaction, and then from there it goes into the citric acid cycle, where all of the carbons of the 6-carbon molecule, which is now 2 3-carbon molecules, are removed and exhaled if you will, as carbon dioxide. Also, all of the hydrogens and oxygens are pulled off of this molecule, because remember, glucose is C6H12O6. So we have 6 carbons, 12 hydrogens, and 6 oxygens. So all the hydrogens are pulled off, and they contribute to making this hydrogen ion concentration gradient across the cristae of the mitochondrion. And it's this hydrogen ion concentration that powers ATP synthase. It basically forces the movement because we just talked about diffusion, and this is what allows it. It's facilitated diffusion of the hydrogen ions down the concentration gradient, and this basically turns the turbine of ATP synthase that allows a phosphate to be put onto ADP. It's pretty awesome. And the output here is from 1 glucose is 36 ATP. And then we get to anaerobic glycolysis. If you've ever done any type of exercise and felt like you're going to die, which is usually every time because there's no oxygen at elevation, anaerobic glycolysis occurs with the absence of oxygen. Now, if you think about it, your cells are never fully without oxygen. There might be a slight oxygen debt at one point, but they're not ever going to be long without oxygen, otherwise they die. So, what animal cells do when there's not enough oxygen to move into or to help the pyruvate in the mitochondrion be broken down, what happens is this then turns into lactate. And lactate, or lactic acid, as we so know from when our muscles ache after a good workout, is then, when there's more oxygen, is then reconverted back into pyruvate and goes back into the mitochondrion where it can make more ATP. But in the instance, we need some ATP, so basically just does lactic acid fermentation. So now at the end, you should be able to do the following things. Apply the definition of diffusion and osmosis to concentration differences across the membrane. And with this, understand tonicity. Understand if you put a cell in a hypertonic solution or a hypotonic solution, what's going to happen to the cell and where the water is going to go. Determine between the various types of active and passive transport. Define endocytosis and exocytosis and relate to functions within the cell. And know the 3 different phases of cellular respiration and where each occurs. Glycolysis is the only one that occurs in the cytoplasm. Everything else is in the mitochondria. Thanks for joining me. This has been another wonderful episode of Human Biology with Dr. B. See you next time.